Check your patient identifiers and put a matching label on a 50 ml tube. Gently mix your sample by inverting the tube a couple of times, checking for clots. Transfer your sample to the 50 ml tube. The max volume of sample we can use is 4.5 mL. Make sure to jot down the sample volume on your setup sheet, rounding to the nearest half mil. Due to the fragile nature and sometimes limited concentration of plasma cells, you want to be extra gentle handling the samples throughout this process. Next, dilute your sample 5 to 10-fold using DPBS that doesn't contain calcium or magnesium. You can use the table to figure out the volume of PBS to add. If you're setting up multiple samples, you can try to use similar volumes of PBS within the individual sample ranges. Mix the sample gently by pipetting up and down or swirling within the tube. Centrifuge the sample at 300 G for 10 minutes without the brake on. This is program 5 for the centrifuge on the fish bench. Because the brake is off, this step will take roughly 30 minutes. During this time, you can thaw the DNA Swin solution to room temperature if your sample is over 24 hours old. After you spin your sample down, set the vacuum to a slow aspiration speed and discard the plasma, careful not to disturb the buffy coat or red blood cell pellet. Ideally, the volume will be close to or a little less than the original sample volume. For samples not drawn earlier that day, add DNA Swin solution and gently swirl the sample. This will help reduce cell clumping. Next, label a 14 milliliter tube. I find it also helpful to write small lines with a Sharpie on the tube at 10 milliliters, the original sample volume, and double the sample volume. Transfer your sample into the 14 ml tube. If it's less than the original volume, you can resuspend it with DPBS, rinsing out the prior tube. Next, add equal parts EasySep red blood cell lysis buffer to the 14 ml tube with your sample. You can use the buffer to rinse out the 50 ml tube if you didn't in the prior step. Gently mix your sample up and down 2 to 3 times. Next, add the positive selection cocktail to the sample. This cocktail should be colorless. Set your pipette to the correct volume using the table. Add the cocktail and gently mix it up and down 2 to 3 times. You can cap your sample if you want to. Set a timer for 3 minutes. Next, you will be adding the EasySep dextrin rapid spheres. This cocktail has a brownish tint and appears to have a coffee grain sediment at the bottom when unmixed. To ensure proper distribution of particles, vortex the rapid spheres for 30 seconds. Once your timer runs out, uncap your sample and add the rapid spheres using the same volume as the positive selection cocktail. Again, gently mix your sample and set a timer for 3 minutes. Next, we need to top off our sample using the regular EasySep buffer, which is different than the lysis buffer we used earlier. Use the table to figure out how much buffer to add. For diluted samples over 2.5 milliliters, I suggest pouring 35 to 40 milliliters into a 50 milliliter tube and using a transfer pipette to add it to your sample. Once you've added your buffer, mix your sample gently and place it into the magnet uncapped. Also be sure that the magnet is in a spot accessible to the vacuum because you don't want to move the magnet once you put your tubes inside. Press firmly against the bottom of the tube to make sure it is snug against the back of the magnet. Set your timer for 10 minutes. Once your 10 minutes is up, put a clean pipette on the aspirator and set the vacuum to a slow aspiration. Leaving the tube in the magnet, slowly take off the supernatant until you hit the sludge, around 1 to 2 milliliters. Take the tube out of the magnet. You should see a brown streak of cells on the back of the tube. Add buffer back up to the desired volume, rinsing off the cells from the back wall and gently mixing. Place the tube without the cap back in, making sure to push the base of it back against the magnet. Set your timer for 5 minutes. Repeating the previous step, place a clean pipette onto the aspirator and gently take off the supernatant. This time you want to go to the bottom of the tube, taking off almost all the supernatant and any clotted material. Take the tube out of the magnet. Add buffer up to the desired volume, rinsing the cells. Gently mix and then place the tube back into the magnet. Set your timer for your final 5-minute incubation. After 5 minutes, take off the supernatant going all the way to the bottom of the tube. Take the tube out of the magnet and rinse the tube again with buffer. Transfer the sample into a 15 milliliter centrifuge tube, bringing the sample up to the desired volume. You can use any remaining buffer to rinse out the 14 ml tube. Place the tube in the centrifuge for 10 minutes at 1000 RPM with the brake on. This should be setting 3 on the centrifuge on the fish bench. When the sample is done spinning, you can go straight into harvest.